Protein synthesis and secretion is a vital cellular process that ensures the production and delivery of proteins to their designated locations. This process, integral to cellular function, involves several well-organized steps where various cellular components work in harmony. The journey of protein synthesis initiates in the cell's nucleus. Here, the genetic blueprint residing within the DNA is transcribed into a messenger RNA, mRNA molecule. This transcription process is crucial, as mRNA serves as a temporary copy of the genetic instructions necessary for creating proteins. Once synthesized, the mRNA exits the nucleus through nuclear pores and enters the cytoplasm, where the ribosomes await. Ribosomes, either freely floating within the cytoplasm or attached to the endoplasmic reticulum, play a pivotal role in translating the mRNA into a polypeptide chain. Each series of 3 nucleotides, called a codon on the mRNA sequence, corresponds to a specific amino acid. Transfer RNA, tRNA, brings the appropriate amino acids to the ribosome, ensuring the correct sequence is followed, as dictated by the mRNA template. Adjacent to the ribosomes, the endoplasmic reticulum, ER, acts as a critical site for protein folding and processing. The ER is divided into 2 sections: the rough ER, studded with ribosomes, and the smooth ER, which lacks ribosomes. As the nascent protein emerges from the ribosome, it enters the lumen of the rough ER, where it undergoes folding and modification. Chaperone proteins assist in achieving the correct conformation, while enzymes may add carbohydrate groups in a process known as glycosylation. Once the proteins are correctly folded and modified, they are packaged into vesicles and shuttled to the Golgi apparatus. The Golgi apparatus functions as a molecular warehouse, further modifying the proteins as they move through its stacked cisternae. It is here that proteins receive any additional modifications necessary for their specific functions, such as phosphate groups or further glycosylation. After processing, proteins are packed into vesicles once again and directed towards their final destinations. Some proteins are targeted to the cell membrane, where they may function as receptors or channels. Others are sent to organelles like lysosomes, where they participate in the breakdown of cellular waste. In cases where proteins are secreted out of the cell, such as hormones or enzymes, the vesicles merge with the plasma membrane in a process known as exocytosis. The protein cargo is thus released into the extracellular space, where it can perform its designated role in the organism. In summary, protein synthesis and secretion is a complex, yet fascinating process that is essential for cellular and organismal function. Every step, from transcription in the nucleus to secretion at the plasma membrane, highlights the intricate choreography of cellular machinery working together to maintain life.